I am Ms. Sloan, and this is my excellent APBio 7th period support. Yay! Can I show them you? Can I show them? Look, here they are. Look at them. Look at them. I love them. I love them. You see them? Okay, I love them. Okay. Now, okay, so you're in if you want to be in, yes? Okay. So for chapter 13, I need to make this smaller. Wish I was a baller. Okay. Chapter 13, regulation of gene expression. Here is the list, okay, of the different topics. So look through, differentiate between and provide authentic examples of inducible versus repressible operons, positive versus negative control, prokaryotic versus eukaryotic control of gene expression. Part of this kind of bleeds into number 2. Name, describe, and differentiate between the 5 levels of control in eukaryotes. This I pulled right off the College Board, the thing, the unit master hyperdocs that I have, and that's pulled from the College Board. Mutations are the primary source of variation, right? Once you have those mutations, how can you can get new combinations through sexual recombination, but do bacteria typically have sex? No, they don't. Can they? Yes, but that's a different unit. We're going to talk about that tomorrow. Whether a mutation is detrimental, beneficial, or neutral depends on the environmental context. Can you think of examples of each? We have talked about that. Think about something that is neutral right off the bat. See if you can remember, if that helps you. What do you say? Silent mutations, right? So that's a mutation, but it neither helped nor hurt, right? Does that make sense? And then a mutation that's detrimental, you know a ton of those, right? I heard it. What was that? Cancer is a good one for you, right? If a proto-oncogene mutates to an oncogene, we've looked at what kind of mutations? Point mutations and the diseases you can have from that. And then what's the other one? Frame shift mutations, right? Those are all detrimental. Beneficial adaptation. If you're a giraffe and your neck gets longer, would that be a benefit? Yeah, because you get to eat more leaves. So I'm just telling you, we want to start pulling things together from previous units so we don't think of mutations in isolation. That's why I re-put in the slides about cancer, so you would think about that as well within this chapter. Okay, and differentiate between point and frame shift mutations, and have you done your highly suggested reading and thinking? All right, are we good on that? Okay, so this is the basic premise of prokaryotic control of gene expression. These are your players. It doesn't always work like this because we've talked about inducible versus repressible operons in this situation. But the part that remains the same is regulators always code for what? Repressors, every time, right? Regulators code for repressors. What could you say about operators every time? Repressors bind to them. That is a job of an operator, right? A repressor may or may not bind to it. What's the job of a promoter? RNA polymerase binds to the promoter. And then you don't start the actual transcription until you reach the structural genes, right? Whatever the genes are in question. Okay, that part remains the same. Whether a repressor is born ready and starts repressing right from the start, and you have to induce it to come away and get off the operator, or the repressor doesn't get on the operator unless it has a co-repressor, that's different. It's just that repressors repress, right? If they're active, that's what they'll do. So that's the basic premise. There are any questions on that you need me to answer? Okay. So then we talked about that there are two basic strategies. I don't know where to put myself. That is clearly better. How about over here? Okay, whoever you're sitting next to, if you're in the slate seat, can you tell them strategy 1 and see if you can own that? And if you're in the blue seat, tell them strategy 2. No, cancel. Cancel. It's going to seize now. It's just not going to be my day. It's coming. Hang on. There we go. Okay. I hope you're done. I want to keep going. Whenever you're dealing with a repressor, what kind of control are you dealing with? Negative control, because repressors repress. Okay, in this first scenario, I will make these proteins until I have enough of them and then shut this operon off. So it is on and until it's turned off by the product. That product can then be a what? Remember? What will that product become? A co-repressor. Nice. A co-repressor. Who will then, you have enough of that product, and that's very typical of what kind? This or this? What do you think this, what kind of pathway is this? Anabolic pathway. So you've built enough of it, so when you have enough, you have feedback, right? And that product that you formed acts as a co-repressor, which then activates the repressor and turns the whole thing off, right? Turns off that pathway. And remember, on these scenarios, when you have operons in prokaryotic cells, everything you need for building that tryptophan, it's in sequence, yes? Right? So it's controlled by that one operon. Is that different in eukaryotes? Yeah, in eukaryotes, there could be genes in several different chromosomes. All right? Then strategy number 2, I will make the proteins when I need them and turn the operon on. So it is off until I need it. And then you have, so the repressor is active. And when you have an inducer, right, like lactose is the example we looked at, if you don't have any lactose to digest, you don't need those enzymes. And the reason why I'm walking you through this is when you take your AP exam, whether it's mine or the one that you take in May, they will give you scenarios and say, what's the operon doing now? And they'll give you several different scenarios. So you need to be able to analyze the why of it, okay? And so in this case, I'm not going to turn it on until I need it, because this is typical of what kind of metabolic pathway? Catabolic, where you're breaking something down. Now I have arabinose, I'm going to turn on the genes to digest arabinose. Now I have lactose, I'm going to turn on the genes to digest lactose. So those are your two. Has everybody signed the sign-in sheet? So it can just go in the back. Thank you, Corbin. Corbin, I saw you in walking class walking over there. Did you walk to Starbucks? No. Walk over there. Oh, okay. All right. So those are your two strategies. And then here are the slides to go over that. So in this one, okay, you have an inactive repressor. So the RNA polymerase can get on the promoter. Yay. You're going to transcribe the enzymes you need to build what amino acid? Tryptophan. If you have enough tryptophan itself acts as a what? Co-repressor. Then it activates the repressor. It gets on the operator. Now RNA polymerase cannot get on the promoter. I have enough of it. When I run out of tryptophan, if I were a bacterium, if I run out of tryptophan, I don't have any more co-repressor. So therefore, what's going to happen? The repressor is going to get off the operator, and those genes are transcribed. All right, we good? Are you strong? Okay. Then the second scenario was with the second scenario, your repressor is sitting on the operator, and it's going to stay on that operator until you have something to take it away. You won't need to take it away until what is present in this scenario? Lactose. When lactose binds to this active repressor, the lactose acts as an inducer. That's why it's called an inducible operon, pulls the repressor away. But we know more to the story, right? Because RNA polymerase still won't bind to the promoter unless what? Transcription factor. You remember the name of the transcription factor? CAP. What makes CAP start helping RNA polymerase bind to the promoter? Cyclic AMP. Boom shakalaka. And why would I have so much cyclic AMP? because I have no glucose. So when glucose levels are low, cyclic AMP is high. Cyclic AMP binds to CAP. CAP helps RNA polymerase bind to the promoter. So you have two conditions to meet before you start digesting lactose. Are you okay with that? And here is just one of those conditions met. Lactose is here acting as the inducer. That's why it's called an inducible operon. But we know we need the transcription factor there. There is a CAP binding site as well. Yes, and I have slides that show that, I think.
Is this operator
operon?
Operon a negative and positive?
Yes, you can see both control mechanisms in this one, both negative and positive. Okay, so then here you can see, I think it's the next slide. I can't remember. I put this together a little bit ago. Okay, yeah, here you go. So this is showing you the positive control. Here's cyclic AMP. Cyclic AMP is only there when my glucose levels are low. That's a cell signal that go ahead and start making the enzymes to digest lactose because it has a preference for glucose over lactose. So it has to have both a combination of lactose and no glucose to get these genes transcribed. Did it go? Oh, okay. And what's happening here? What can you tell me about this scenario? There's no. Is there lactose present?
Yes.
Yeah, there is lactose present. How do you know lactose is present? No repressor, and RNA polymerase is trying to bind to the, but your tell is that there is no repressor. If you had a repressor on here, that would mean that there was no lactose to remove it. Right? We good on that? But is this gene being transcribed? No, because it's not got a good bind onto the promoter. It needs a little help from a transcription factor, and that transcription factor is inactive. He needs to be activated by what? Cyclic AMP. So CAP needs that, he needs to be induced. It's still inducible. It needs to be induced by the presence of cyclic AMP. And these structural genes need to be induced by the presence of lactose. Are you okay? All right, and then all those scenarios are put together here on this slide. Okay, so we have a CAP binding site. We have where RNA polymerase binds. We have our operator, and here's our gene coding for the digestion of lactose. In this case, glucose is present. If glucose is present, definitely CAP is not around, right? No CAP. Okay, lactose is present. We can tell because there's no repressor on there, right? But RNA polymerase isn't binding because it's not getting any what? Help. Are you okay with that? Okay, work your way through the remaining ones with your bio buddy and see if you can explain it instead of me explaining it, okay? Because I've already done it once and I want to make sure you understand it and then you can ask me questions. All right. Any one of those you want me to go over? I'll go over any one you want. You good? Okay. So then let's transition to control of eukaryotic control of gene expression. So here are your 5 biggies, right? And I have slides to help you go through all 5. But the first one, let's see if we can just talk about it a little bit. The first one is the availability of what? DNA, right? The availability of the DNA. What do you call it when we put our DNA tightly coiled and put away? Heterochromatin. When it's available, it's, what's another way to switch off some DNA? Starts with an M. Methylation. If you have a methyl group, it'll usually shut it down. Remember, we talked about epigenetic factors controlling whether or not that DNA is available. And do you remember how I talked to you about histone proteins forming the nucleosome and the DNA winds on? If it's not a histone protein, it's called non-histone proteins. And they do affect what DNA is transcribed and translated. Histone proteins are part of nucleosomes. They're purely structural in nature, right? They're just winding so you can wind the DNA around. Non-histone proteins inside your nucleus are going to regulate what DNA is unwound and or wound up. Okay, when we look at transcriptional control, it's all about, can RNA polymerase bind to the promoter? We transition, and that has to do mostly in eukaryotes with what kind of control? Positive control, right? As opposed to prokaryotes. Prokaryotes, you have an operon, and all the metabolic pathway is in a line right after that one operon, and you can control it with one operator. Do we function like that with all of our DNA? No, it's all over the place. So we have multiple, as long as we have the same conditions inside the new, right? If we have those same conditions, everything we need, transcription factors, transcription activators, if they're all there, then we can turn it on those genes on all the different chromosomes. Post-transcriptional control is all about what? Cap, tail, and yeah, you're doing those spliceosomes and you're cutting out your introns, yes? Now, those intragenic, right? As we learned, intragenic sequences, we're removing those. And when you do that, all of these, chromatin transcriptional and post-transcriptional, you're still inside the what? Nucleus. It also includes your ability for that mRNA to leave the nucleus as well. Because you could be all cap-tailed and cut, but if you cannot get out of the nuclear envelope, you will not be translated, right? And then translational control is leaving the nucleus and getting on that ribosome and having your codons read. That's all translational. Post-translational, we're not dealing with any nucleic acids. What are we dealing with then? Proteins. It's all protein at that point. That's the only one of all the levels of control, the only one where you're talking about the protein product. All right, so then I put together a few slides for you on that. There we go. So on eukaryotic regulation, chromatin structure, we know about the histone proteins. Remember we talked about whereas methyl close it down, usually acetyl groups do what? Open up. Yeah, makes it active acetyl. We talked about evidence of DNA that is euchromatin are those lamp brush chromosomes where they are available. Okay, then when we go to epigenetic factors, yes, we can talk about chromatin availability, but I just want to remind you those epigenetic factors is anything above the DNA that it's influencing what proteins we have. So epigenetic factors do affect things in the nucleus, but out in the cytosol as well. Okay, it's still epigenetic if it's above, right, above the DNA itself, above the code. Go. There we go. We talked about examples when we talked about bar bodies. That's specifically the heterochromatin of what? Females, but be more specific, it's the second X chromosome, right? That's what a bar body is. So you don't refer to bar bodies of other chromosomes, but right, you might have areas that are heterochromatin. Maybe it's the whole one, but if it's a bar body, it's specific, the second X in females. And we talked about identical twins having different color eyes as long as those twins are female, right? Because it depends on what X is getting expressed. Females are mosaic in that way, whereas males have one X and one shot at those traits. Are you okay with that? All right, moving on. Okay, transposomes, jumping genes. I bring this up here because DNA that was there that you thought maybe I could transcribe and translate and it was associated with the promoter, it could get up and jump somewhere else. Yes? Okay. All right. And then transcriptional control, we talked about that being the most important factor, and something that was, everything is the same, what we learned in prokaryotes, except we're leveling up. We now have transcription activators that bind to a what? Enhancer. And those activators bound to the enhancer, you have this hairpin loop that brings the transcription factor where it needs to bind on. Okay. And then we gave an example. I don't know what the next slide. Hang on a second. Yeah, the data box. Remember that? And we talked about that being part of the what? Promoter. Part of the promoter, right? And that can be universal. It's a conserved sequence to help and facilitate transcription. All right, then we talked about post-transcriptional control. Any second, there we go. Post-transcriptional control, this is why your proteome is so much larger than your genome, depending on what exons you keep and what get cut out. And you just need to remember it's cap, tail, exons, and your ability to leave the nucleus. I take this opportunity because these small interfering RNAs are cut out in post-transcriptional control, right? Like sections of RNA could be cut out. They can influence later on translational control. So as a result of post-transcriptional control, you create these RNA products which can go mess with your translation later. So that's why I talk about them in two places. Does that make sense? Okay. And so there's a lot of different RNAs. And are all of them bad? No, they can be good too. They can be regulatory. All right. And then translational control, this can be, you can have interference with those small interfering RNAs, but it's your ability to get onto the ribosome and build your protein. Post-translational control is the folding. And so we know, who do we need help from to fold correctly? Chaperones. Do they help you fold it into this secondary structure? No. What does the secondary structure do to the amino and the carboxyl groups, right? And that interaction. And what are the two types of secondary structures? Alpha helix and perfect. Okay, just checking. Good. Then we talked about how can it be messed up. So the first one we talked about was point mutations. Point mutations, you can point to a nucleotide that caused the problem. If the DNA is messed up, then it's possible that the codon is messed up that's transcribed from it. Does it always mess it up? Why not? Yeah, the code is degenerate and you have redundancies, right? More than one way to skin a cat. There's more, I'm not saying you should skin a cat, but there's more than one way to get that same amino acid. So it's called a silent mutation, right? If it doesn't change the sequence of amino acids. If you get a stop codon, how many stop codons are there? How many start codons are there? One, good. If you get a stop codon, it's called what? Nonsense. I'm kind of covering it up there with my face. So remember, I remember that like non-stop, no. Okay, it's a stop codon. It's a missense, a mistake if you get any other amino acid from that codon, anything that's not a stop codon. Then we totally went all over frame shift mutations. And what you need to be able to see is if on a quiz or a test, I say there's a deletion, I want to ask what's the new types of, what codons would I expect? Just like I showed you here, the new codon is now THB, right? So make sure you would understand if I say, oh, I added a nucleotide at the front, be able to block it into the next sets of 3 codons. Questions about that? It's easy. It's just I would give you the basis. I just taught it with you this way. Okay. And then we talked a little bit about proto-oncogenes and tumor suppressor genes. So those are mutations. We've already talked about that before. So I'm going to let you review that if you need to. And then the last thing we talked about was the Ames test. And remember with the Ames test, in order to see if something would cause a mutation, you start with broken bacteria already that can't produce the amino acids they need. And then you expose your chemical that you're testing. And if it corrects them to the ability to make that protein and they can grow on what's called minimal medium, then you know that it is a mutagen. Even though it's doing a good thing by correcting it, it did in fact change the DNA. So if it changes it from B to A, you know it could also probably change it from A to B. Good. Okay, and then that, my friends, was it. Any questions? Yes.
What do the proteomes and the lysosomes do in post-translational?
Lysosomes remember, they do the digestion. So if you have a sequence, a nucleic acid, they can break it down. All right. Good job, you guys.
I love my notes.